Hello students, I'm RP Jaiswal, Assistant Professor at School of Medical and Allied Science, GT Goenka University, Haryana. We learned clinical enzymology, endocrinology and toxicology course today. And here we learn about introduction to enzymes, classification of enzymes and lock and key model and induced feed model theories. Let's talk about introduction to enzymes. Enzymes are proteins that increases the rate of reaction by lowering the energy of activation. Enzymes are also called biocatalysts, and here this enzyme catalyzes nearly all chemical reaction taking place in the cells of the body. Enzymes are not altered, they are not consumed during the reaction, and enzymes are also reusable, means they are used after completing the reactions. Importance of enzymes. Enzymes play very important role in digestion, metabolism, diagnosis and therapeutic process, and all biochemical reactions are enzyme catalyzed reactions in living organisms. So if you talk about digestion, so digestion of the food require enzymes. In the case metabolic process, that is anabolic, catabolic, it also require enzymes. Even diagnosis of enzymes, as a reagent we uses enzymes, and even some cases therapeutical process, the treatment process, we use enzyme as a drugs or medicine to treat some of the disorder. So level of enzymes in blood are diagnostic importance. It is a good indicator in disease such as myocardial infarction. So if you are able to estimate the level of enzyme in the blood, you can be able to diagnose myocardial infarction. Enzymes can be used therapeutically, such as diagnostic enzymes, which can be given orally to treat the patient suffering from digestion related disorder. These days enzymes are used in lab procedure for estimation of blood analytes such as glucose, cholesterol, drugs, antigen, hormones. So enzymes are also used as a reagents in the estimation of these analytes. We'll talk about active sites. What is active site? Let's know. So in this diagram we can see where is active site. Yeah, let's see the structure of the enzyme. The large structure is the enzyme, and there is a typical pocket. Here enzyme molecules contain a special pocket or cleft called the active site. So this is what we define the active site. It's like a pocket or the cleft on the enzyme, and another molecule, the substrate, which is yellow color, which has a similar structure to the active site, are going to bind by entering the active site of the enzyme. After that, the next structure we can see the enzyme and the substrate has made a complex. The enzyme changes shape slightly as substrate binds. So according to the shape of the substrate, there is a change in the active site's conformation. After that, the enzyme catalyzes the reaction. We can see the changes in the color of the substrate here. We got now the new compounds, that is called the product. So we got enzyme product complex, and the last, the product which has formed is leaving from the active site of the enzyme. So products are the two small pieces that we have got from the large molecule, that's the substrate. We are going to know about the example of degradation reaction that occurs in our body. Here we see the red color structure, that is enzyme, the yellow color structure, that is the substrate, or we can say the food particle. So this food particle is a large molecules which will bind to the active site of the enzyme, and after that we see the combined enzyme and substrate. So a substrate has bound to the active site in a proper orientation, and at the end we can see the product, which is two small pieces that we got from the large piece substrate, and again now enzyme is free here. So substrate is converted to product, the small molecules that we have got at the end. So here we studied how enzymes break down food into the nutrients in the degradation reaction. Let's talk about the synthesis reactions which takes place with the help of enzymes. Here again we see the enzyme which is red color substance and red color structure, and another is the yellow color substrate which is small molecules in 2 in number. So this is small molecules which is a substrate which will bind with the active site of the enzyme. Now you can see the combined enzyme and substrate has formed, and at the end you can see the product which is the bigger molecules as a product which has been uh, detached from the enzyme active site. Enzyme is again free to bind another reactions. So this kind of enzyme is a synthetic enzyme which synthesizes new molecules or larger molecules from the small molecules. Here active sites. Now classification of enzymes. Enzymes are classified in different way from the ancient time to the current time. Earlier, let's talk about the nomenclature. So tribal name means local name which gives no idea of source, function or reaction catalyzed by the enzyme. So locally the enzymes were named. Nomenclature means naming. So naming of the enzymes which were given the local name, so it has no any idea about the source, function and the reaction catalyzed. For example, trypsin, thrombin, pepsin. So these are the local name, and it doesn't give about the what kind of reactions are catalyzed, what is the source, what is the type of substrate which are going to be converted into the product. Most enzymes names end in ASE. So at the last of the enzyme name there is ASE, like nuclease, lipase. These are the again the you can say the tribal name. So there were the confusion that by knowing the name of the enzyme, we are not able to know the source, function, and reactions, and this tribal name was somehow confusing. Systematic name. So now what happened? The systematic name, the new name was given, or systematic name is also called a scientific name. So the International Union of Biochemistry initiated standards of enzyme nomenclature which recommended that enzymes names indicate both the substrate acted upon and the type of reaction catalyzed. Here from the enzymes names we can also know that the type of reaction and the substrate that is going to be acted on. So this is a scientific name. According to IUB, that is International Union of Biochemistry, an enzyme has two parts, first part and the second part. First part is the name of the substrate for the enzyme, and the second part is the type of reaction catalyzed by the enzyme. This part ends with the suffix ASC. For example, lactate dehydrogenase. So lactate is the first part, which means substrate for the enzyme, and the second part is the dehydrogenase, which is the type which indicate the type of reaction catalyzed, like dehydrogenation reaction or oxidation reaction. And you can see suffix ASC in the last part of the enzyme nomenclature. Let's talk about the classification of enzymes in detail. This International Union of Biochemistry has classified enzyme into 6 major classes. Oxidoreductase, which means catalyzing oxidation reaction. Transferase, which means catalyzing group transfer. Hydrolysis means catalyzing hydrolytic breakdown. Lyase, catalyzing removal of groups by mechanism other than hydrolysis and leaving behind double bonds. Isomerase means catalyzing conversion of isomers, and the last ligase, which means catalyzing formation of bonds and new compounds. So these are the different class and its introduction. Here we'll know about some of the example of each class of the enzyme and how the reactions are catalyzed by each class of the enzymes. This is oxidoreductase, for example, called dehydrogenase. cytochrome oxidase, L-D-amino acid oxidase. And type of reaction catalyzed is oxidation reduction reaction. You can see AS2 is a compound and B is another compound. So AS2 is reduced form and B is oxidized form. So oxidation of AS2 become A and reduction of B become BS2. Next class is transferase, where examples are hexokinase, transaminase, transmethylase, phosphorylase, and this catalyzes group transfer reactions. So X is the group on A, and this is the one compound, and B is another compound. So X group from A will be transferred to the B compound. So we got A plus BX. So B is a compound now having X group. Third class of enzyme is hydrolase. Its examples are ligase or lipase. Sorry, it's a lipase, cholinesterase, acid alkaline phosphatase, pepsin, urease. And these examples comes under the hydrolase, and this catalyzes hydrolysis reactions. Here we see the AB is a big compound, and this is going to be hydrolyzed or broken down with the help of water. Water contains hydrogen and hydroxyl group. So when this AB compound is broken down, so A get H and B get BOH, and now we have two small pieces of AH and BOH from we got from the AB compound. Lyase, example aldolase, fumarase, histidase. These are the common example which comes under the lyase, and this catalyzes addition elimination reactions. Here AB is a one compound, XY is another compound. So these two compounds are joined together, and we see that A binds with X become X and B binds with Y become BY. So AB and XY join together to produce AXBY compound, single compound. Isomerase. Here examples are triose, phosphate isomerase, ketoisomerase, retinol isomerase, phosphohexose isomerase. And this isomerase type class of enzyme causes the interconversion of isomers, like A become A dash. So there is no change in the molecular formula, just there is a change in the structure because of the changing in the position of hydrogen or hydroxyl group from one carbon to other carbon. Ligase is the last class of the enzymes classification, where examples are glutamine synthetase, acetyl coenzyme A carboxylase, succinate thiokinase. And ligase catalyzes condensation reaction, usually dependent on ATP. So A plus B, A is another compound, B is another compound. So A and B will be joined together and it will make AB. So it's similar to how we can see lyase, but here we see the reaction is dependent on ATP. So ATP breakdown releases energy which helps to join AB to AB. So here we studied classification of enzymes. EC number. What is EC number? Let's talk about this. Classification of enzymes does not take into account based on amino acid sequence or protein structure. So this is about the scientific classification we are talking about. There were confusion that how enzymes should be classified. So the enzymes were classified based on the chemical reaction, the type of chemical reactions, not on the account of amino acid sequence or protein structure of the enzymes. The nomenclature of each enzyme was determined by the Enzyme Commission in 1961, and then it was updated in 1992. Hence, all enzymes were assigned their EC number. The code was given. This Enzyme Commission code or enzyme code was given in 1992. Finally, so each enzyme has got a number, and EC number has a 4 digits: A, B, C. For example, first digit A indicate class, means there are 6 class of enzymes, and out of this 6 class, to which class this A digit belong. So EC number has a, basically when you talk about EC number, there are 4 digits in the EC number, and the first digit indicate the class, the second digit, that is B, indicate subclass, and the third digit indicates subclass, and the D, that is the fourth digit, which indicates the enzyme number finally. So let's see that BC describe reaction type, and while indicate the distinguish between different enzymes of the same function based on the actual substrate in the reaction. Like B is the number we say, D is the number we say. Example, alcohol NAD oxidoreductase is the one enzyme, and its EC number is 1.1.1.1. So 1 means this is the first digit indicate class of the enzyme, and the second, third indicate the subclass, and subclass, and the last number, that is a enzyme number. So under the subclass 1, the first enzyme is the alcohol NAD oxidoreductase. So first last digit indicate the enzyme number under the subclass. Lock and key theory and induced fit theory. These theories explain specificity of the enzyme action. General introduction to this enzyme specificity. So lock and key theory means where the enzyme active site is complementary in conformation to the substrate, so that enzyme and substrate recognize each other. This is how we talk about the lock and key, that key and lock has a similar structure. Because of that, one lock can be opened by one specific key. So this lock and key theory, what it indicates, that the active site is very complementary in conformation to the substrate. Enzyme active site and the substrate structure are very complementary to each other. Next, induced fit theory, where it indicates that the enzyme active site changes their shape on binding to the substrate, so that the conformation of the substrate and the enzyme active site become complementary only after binding. So lock and key model were first postulated in 1894 by Emil Fischer, and here let us know how it explains how we explain lock and key model. Enzyme structure, we can see this blue color. Here is the active site. You can see the structure of the active site, and here is another compound, that is substrate, which will bind to the enzyme active site. So substrate and enzyme will bind because substrate and enzyme active site has a similar structure or conformation, and so substrate will fit into the active site and we can get enzyme substrate complex. So in the lock and key model of the enzymes, what we have understand that the active site is a has a rigid shape. There is no change in the shape after binding or before binding. It's a rigid shape, and only substrate with the matching shape can fit. The substrate is a key that fits the lock of the active site. There is no change in the active site before and after chemical reaction. This is an older model, however, it does not work for all of the enzymes. This is what the another image is, which says that key, which is a substrate, and the lock, which is an enzyme, which can fit and becomes a lock and key complex. Another image we can see the substrate is the key, so which is going to bind to the active site of the enzyme, and we can see the enzyme substrate complex. Induced fit model, which was postulated by Daniel E. Cosland in 1958, and he is the person who proposed this model. And here we'll see the image that in this fit model, the enzyme active site, the structure and the substrate structure, so both are not similar to each other, but even though substrate has bound to the active site and has made the enzyme substrate complex. In the induced fit model of the enzyme reaction, the active site is flexible, we can see, and it's not rigid. The shape of the enzyme active site and substrate adjust to maximize the to fit the shape, which improves catalysis. There is a greater range of substrate specificity, means different group of substrate can bind to the particular enzyme. This model is more consistent with a wider range of enzymes. For example, if you see that hexokinase is an enzyme, and hexokinase can bind all hexo sugar. like all monosaccharide in hexosugar, like glucose, fructose, galactose, but glucokinase can bind only glucose. These are the another explanation for Induced theory, where reactants approach enzymes and reactions binds to the enzyme changes shape, and we got a chemical reaction, biochemical reaction, and the reactants will become product now. Thank you so much for listening.